Hello everyone, and welcome to the redo of the last slide in your prep screencast. So I want to basically start all over again so that everything is clear for you. So whenever we get some kind of stimulus, whether, I don't know, it's, let's say, hot coffee pouring on you, or you just putting your clothes on, or seeing a beautiful flower outside, whatever stimulus it is, visual, auditory, pressure on your skin, all of that is going to be a stimulus that's picked up from your receptors, okay? And those receptors, let's put this up here. Those receptors connect to a sensory neuron. Remember, what's my other name for, I'm not even spelling this right, am I? Yes, I was. Okay. What's our other word for sensory neuron? Afferent. Yes. So sensory, so I put an S there, neuron or afferent neuron is bringing that information into our integration center, which would be our brain and spinal cord. And within this area, we would have another nerve cell, and that would be an interneuron. Okay? And from there, we transmit this information out through a motor neuron or efferent neuron to the effector organ, right? So the way that we're getting the stimulus information is picked up by this receptor and is sent through this neuron, or this impulse is being sent through the interneuron and then through the motor neuron. So what I want you to understand is, here is my nerve cell. Okay, this is the dendrites, this is my cell body, the nucleus. Here's my axon with the axon terminals. So information is being transmitted through here. So the site where this action potential is occurring is, let's say, right here. Okay, so now let's take a closer look at this. We're going to look at the plasma membrane of this nerve cell. So if you remember, our plasma membranes, they were made up of a bunch of those phospholipid heads. Remember that fluid mosaic model? It's a semi-permeable membrane. And here we have the phospholipid heads with the tails coming on down on either end. Okay, that's all along here. We're going to have channels located along this axon in its plasma membrane. This is the inside of our nerve cell. Okay, so this is going to be intracellular, this is going to be extracellular. So what I want you to know is every single cell is going to have a resting membrane potential, and this resting membrane potential of our nerve cell is going to be 70 millivolts. Now, what does a volt mean? A volt is just a measure of potential energy. Okay, and this is generated, sorry, not potential energy, potential electricity, which essentially is going to be similar, but I'm not a physics person. So anyway, this is going to be generated by separated charges. Can you even read this? I hope so. Anyway, good thing you can hear me talking, I hope. So the inside of our cell is negative 70 millivolts, like we mentioned. And the outside of our cell is going to be 0 millivolts. So we have a wall, the semi-permeable membrane, that is separating these charges. I'm just going to replug this pen. So what does that mean for us? We have ions in here, and these ions are creating these charges. So let's talk about what we find in each area. Here we are going to have a high concentration of potassium, and we have a high concentration of those negative proteins, which we're going to represent with the letter A. So these proteins are so large and so negative that they are causing this negative 70 millivolt resting membrane potential. Outside of the cell, we have a big concentration of sodium, okay, which is positive. That's fine. So we got positive charges going on here. We also have high concentration of calcium and a high concentration of chlorine. Okay. Now you might be thinking, well, chlorine's negative. Yeah, but we have way more sodium in here than we do chlorine. And same is true for potassium. We have way more positive sodium out here compared to the amount of positive potassium inside the cell. So that's why we have 0 millivolts here at this level and negative 70 millivolts in the resting membrane potential. Great. Now let's talk about this action potential and what are the steps in which this action potential gets generated. So like we said, we need a stimulus, okay? So that stimulus causes our sodium ion channels to open on up and let some of the sodium on the outside in, okay? Before that, so let's talk about like what's going on right here. Let me change colors real quick. Actually, before I do that, let's mark this. So this is what's happening here. We're getting the inside of the cell to change from this negative 70 to move a little bit more positive. What we want to do to have an action potential cause is to raise this to our threshold of negative 55 millivolts. Once it passes this threshold, then we're going to depolarize the cell and gain our action potential. But real quick, let's talk about what's going on before that, because I forgot to mention this real quick. Before that, we have this sodium potassium pump. Do you guys remember this guy? This guy pumps 3 sodiums out of the cell and 2 potassiums in. So we're getting a lot more sodium out here than we're getting potassium in here. That's another reason why the outside of the cell is going to be more positive compared to the inside of the cell. Okay, now that we got that guy out of the way, let's go back to our graph. So what happens when we hit that threshold? After we've had some sodium flood in, well, now we have this like positive feedback loop happening of voltage gated sodium channels all opening up. Our neuron plasma membrane has a ton of these voltage gated sodium channels, and they cause a huge rush of sodium into our cell. What's that going to do to our negative 70 millivolts? It's going to turn positive real fast. So we go from the negative 55 threshold at this point up to positive 40 millivolts, causing our action potential to take place. Now, once it reaches that level, our voltage-gated sodium channels are going to now shut. Sodium channels, this is getting worse and worse as time goes on. close. Okay? And so these sodium channels close, but another channel is going to open. And that is going to be, no, we already did blue. There isn't purple or something. Let's use gray. That is going to be our potassium channels. Our potassium channels are going to open. And when our potassium channels open, that's going to allow, it's allowing our potassium to flood out. Out of the cell. So the reason for the direction in which these ions are flowing is because of it moving from its high concentration gradient to its low concentration gradient. So remember, we had a high concentration of potassium inside the cell, so now potassium is going to want to flow out of the cell. Whereas when we talked about sodium, we had lots of sodium outside of the cell and not so much inside the cell. So that's why it's flowing in this direction inside the cell. So repolarization is what we're going to term when all that potassium is flooding out of the cell. And that is bringing that membrane potential from the positive 40 to negative 70. And sometimes, actually most of the time, we overshoot this or undershoot it, however you want to word it, and we end up at a much more negative level of like negative 90 usually, or negative 100 millivolts. And so this portion, they haven't put it on this graph here, but we can add it in. This is called hyperpolarization. Oh geez. Hyper, like you're really hyper or excessive. Polarization. Okay. And this is also known as the refractory period. That means that it's going to be more difficult to get an action potential to occur for a stimulus to cause the inside of the cell to become more positive again and reach this threshold at this point, because our cell is super negative now. And the reason for that is so that we wait until our inside of the cell becomes negative 70 again, that resting membrane potential via the sodium potassium pump. And this ensures that this area here that our action potential's happening at, it's going to send the impulse in this direction so that we can't go backwards. Our action potential, our impulse will not go back to the cell body. It's going to continue to move in one direction, okay? All right, do I need to mention anything else here? Let's quickly talk about the myelination of our nerve cell. So if you remember that myelin sheath, we had myelin sheaths surrounding some of our nerves, the axon on the nerve, within both our central nervous system and peripheral nervous system. What were the cells that created the myelin sheath in our central nervous system? Oligodendrocytes. What about in our peripheral nervous system? Schwann cells. They're both creating the same exact thing. And in between them, we have these nodes of Ranvier. And these are the sites where action potentials are happening. So an action potential will happen here, and from this area, instead of moving through this entire axon here where we have the myelin sheath, it's going to jump and have the next action potential take place here, and then jump again. so that we can move through this nerve a lot faster. And this is called saltatory conduction. Like I said, this is getting worse as time goes on. That's supposed to say saltatory conduction. Anyway, that about covers action potential. We'll go through this again in class. Meanwhile, if you have any questions before class, I'll be around my desk at 7:30 in the morning. So I will see you tomorrow morning. Take care. Bye-bye.